In the framework of identifying the presence of the rhesus antigen on the red blood cell, experiments were performed as shown in document F on page 118 in your book. The experiment is made up of two sets. This is the first set and this is the second set. In both sets, red blood cells were removed from three different sources. It was removed from a macaque monkey, the rhesus monkey, so called because on the red blood cell we're going to have the Rh antigen. It was removed from the Rh positive individual and from Rh negative individuals, likewise here. Keep in mind that it has been estimated that the majority of individuals are Rh positive, around 85%, while 15% are Rh negative. Apart from removing the red blood cells from three different sources, serum is also removed from rabbit, as observed here. Serum from this rabbit and the serum from this rabbit. The red blood cells removed and the serum are going to be mixed together, and the results are revealed here. In the first set of our experiment, the red blood cell removed from the rhesus monkey Rh positive individual and Rh negative individual showed no agglutination when mixed with the serum originating from the rabbit. In the second set of experiment, the red blood cells were also removed from the three sources and mixed with the serum of the rabbit. But as you would observe here, the results have differed from the first case. As you know, the difference in the result is going to indicate that we have a variable in this experiment which is not found here. Can you identify the variable step done in this experiment that led to a different result? If we look closely at the steps did not differ here. And here the steps did not differ, and here the steps did not differ. The difference is in this step found here, which is not found here. What is the step? The red blood cell of the rhesus monkey were injected to the rabbit before removing the serum, while in this experiment the red blood cells were not injected to the rabbit. So this rabbit here had not been previously exposed to a contact with the red blood cell of the rhesus monkey before removing the serum. In contrast here, this rabbit had been previously in contact with the red blood cell of the rhesus monkey before removing the serum. This led to a difference in the result, as you would see in this case, in contrast to this case. After discussing the conditions and the result of the experiment in detail, let us proceed to do a brief analysis. There was agglutination observed of the red blood cells taken from the rhesus monkey and the rhesus positive individual when mixed with the serum removed from the rabbit that had been previously exposed to a contact with the red blood cell of a rhesus monkey. However, the red blood cells of the rhesus negative individual showed no agglutination. In contrast to experiment 1, as you would recall, where no agglutination happened in all the 3 samples of red blood cell when mixed with the serum taken from the rabbit that has not been exposed to a previous contact with the red blood cell of a rhesus monkey. So how can we explain such results? That is, why did the red blood cells in this case of the rhesus monkey and the Rh positive individual showed agglutination, while here we had no agglutination? As you know by now, when we are talking about serum, we look for antibody, and when we talk about agglutination, this implies that there is a binding between the antibody and the antigens found on the red blood cells. So let's take these notions together and try to explain the results that is revealed in these 2 sets of experiments. Agglutination has happened in this case. That is, anti-Rh antibody should have bound to the Rh antigen found on the red blood cell. Now, where did the anti-Rh antibody come from? Since we are adding serum, this serum must have contained an anti-Rh antibody. In this case here, there is no agglutination. So there is no anti-Rh antibody that had bound to the antigen, that is the Rh antigen found on the red blood cell in these 2 cases. So this serum does not contain anti-Rh antibody, in contrast to the serum that must have contained anti-Rh antibody to bind to the Rh antigen on the red blood cell here and cause agglutination. As you know, it is clear in this case that no agglutination had happened, since the Rh negative individual, the red blood cells of the Rh negative individual has no Rh antibody. That is, the serum here with the anti-Rh antibody is not going to bind on any Rh antigen here, since it is not found. So the question that arises here, how come does this serum contain anti-Rh antibody and this serum doesn't contain anti-Rh antibody? The presence of anti-Rh antibody in the serum of this rabbit can be explained by the fact that this rabbit had been exposed previously to the red blood cell of the Rh monkey that has the Rh antigen. This rabbit, the body of this rabbit, has produced anti-Rh antibody in response to the Rh antigen found on the red blood cell. Therefore, this shows that the serum of the rabbit has anti-Rh antibody which were produced due to the first contact with Rh antigen, and this is going to lead to agglutination here. However, in this case, in the first experiment, this theorem has no anti-Rh antibody, since this rabbit has not been exposed to the antigen before, to the Rh antigen before. So its body is not going to produce any anti-Rh antibody. This is why the theorem has no anti-Rh antibody, and this is why when it was added to the red blood cells of the rhesus monkey and the rhesus positive individual that have rhesus antigen on their red blood cell, no agglutination happened. There was no antibody to bind to the Rh antigen on the red blood cell to cause agglutination. So, in conclusion, the rhesus system is characterized by the presence of the Rh antigen on the membrane of the red blood cell since birth, and the presence of anti-Rh antibody after the first contact with Rh antigen. That is, the anti-Rh antibody could be acquired, I'm stressing on the word could be acquired, during life if the individual is exposed to a blood transfusion in such a way that the transfused RBC, that is the transfused red blood cells, possess the rhesus factor. So, the immune system of the receiver will consider the rhesus factor as an antigen, leading to presence of an immune response where the body is going to secrete or synthesize anti-Rh antibodies. The conclusion to the experiment is that the rabbit serum doesn't contain the anti-Rh antibody except after the first contact, and that such agglutinations are not produced except with injections having the rhesus antigen. How can the concept of Rh positive and Rh negative be applied in blood transfusion? When it comes to blood transfusion, if you are Rh positive, you can get Rh positive or Rh negative red blood cell transfusion. That is, you can get blood from Rh positive individual or Rh negative individual. It would not lead to complications. But people with Rh negative blood should only get Rh negative red blood cells. The Rh antigen, which is found on the red blood cells of an Rh positive individual, will pose a danger for the Rh negative person who lacks the antigen. There are generally no anti-Rh antibody in the blood of an Rh negative person. The anti-Rh antibody will be produced only upon the introduction of the Rh antigen into the blood of an Rh negative individual. When an Rh negative person is given the Rh positive red blood cell, which has Rh antigen, the immune system of the Rh negative individual is going to respond to the Rh antigen and consider it as foreign and produce anti-Rh antibody against the Rh antigen. These antibodies, that is the anti-Rh antibody, will attack the foreign red blood cells, that is the red blood cells coming from the donor, which is Rh positive. The antibodies are going to attack the foreign red blood cell and lead to clumping of these cells or their agglutination, which is going to cause serious complication and can sometimes lead to death. Finally, what are the differences existing between the ABO system and the rhesus system? Since birth, the ABO system, as you have observed, is characterized by the presence of antigen A and antigen B, as well as the presence of anti-A and anti-B antibodies in the plasma. That is, antigen A, antigen B, anti-A, and anti-B are found at birth. On the other hand, the rhesus system is characterized by the presence of the rhesus factor, that is the Rh antigen, at birth, and the presence of the anti-Rh antibody after first contact. This is all for now. Thank you for watching.